Chapter 13 is all about microevolution, and this is the small changes that occur that add up in evolution, and it begins with Darwin's voyage on HMS Beagle. So Darwin went on a voyage. In South America, he went to the Galapagos Islands. These are islands off the coast of Ecuador. So here is Ecuador, and it's on the equator. And this is the equator right here, and the Galapagos are about 600 miles west of the coast of Ecuador. And on those islands, he saw the iguanas on those islands were similar to those on the mainland of Ecuador, but not similar to iguanas on other areas of the Earth's equator. And so this indicated to him that geographic proximity is a better indicator of relationships than is similarity of environment. And this led him to his theory of evolution, where there's descent with modification is the idea. And Darwin had certain influences as well. So Thomas Malthus, had written a short book on competition for resources, and he was hypothesizing that when the human population gets large enough, that there'll be competition for scarce resources. And Darwin wondered if that was the same for natural populations. So that was one influence. Another influence was the geologist Charles Lyell, who had proposed that there's gradual geological change over time. And Darwin saw evidence of this on his trip, and one of those pieces of evidence was that when he was in Chile, there was an earthquake, and he noticed that the ground along the place where the ship had docked had moved up compared before the earthquake and after. The ground had been raised up, and so Darwin realized that as time goes by, the surface of the earth changes. Another influence was Wallace. Now Wallace had reached similar conclusions to Darwin, but somewhat later, about 10 or 15 years later, in a different part of the world. Wallace was in Indonesia, whereas Darwin was most of his conclusions he reached from his work in South America. But Wallace wrote to Darwin, and he, after Darwin came back from his voyage, and he suggested that they co-author a publication, which they did. So next we come to the theory of natural selection. This is the theory of Darwin and Wallace that explains the evolution of species. And this theory can be summarized in 4 points. The first one is that variation exists within a population of a species. So this variation arises from mutations, and those mutations get shuffled around during meiosis. But if you look at populations in nature, there's variation within them. And the textbook gives some examples of that. One is they show some garter snakes. Even though they're the same species, they have many different colors and patterns. And I believe there's a picture of that. Second point is that not all offspring will survive to reproductive age. So many more offspring are born that are going to survive to reproductive age. And so this sets up what's called a survival of the fittest, or a struggle for existence of those variants. And then those variants that are better adapted will leave more offspring. And those offspring will in turn pass on the well-adapted trait to their offspring. So the variation has to be genetic, and it's passed on. And so over time you get a selection for the trait that is more adaptive. So if you look at the DNA sequence of species that are more closely related, their DNA sequence is more similar to species that are more distantly related. So if you were, for example, look at human and ape DNA sequence, it's much more similar than human to, say, a lizard or a snake or something a little more distantly related. And the third is called selection. So mutations that confer resistance to an antibiotic or a pesticide are selected by the application of that antibiotic or pesticide. So the mutations existed before application of these, but they get selected by the presence of those, which kills all the sensitive ones, but the resistant ones that have that mutation that confers resistance then take over the population. And so that demonstrates that selection doesn't in fact occur, and can occur very rapidly in rapidly reproducing organisms such as bacteria. So antibiotic resistance is a big problem in medicine today, and it's a result of the selective pressure that the antibiotic puts on the population, and those that have the mutation, that just happen to have a mutation that confers resistance, then becomes able to thwart the antibiotic treatment. The same thing occurs with agricultural pests like insects. They can evolve resistance to pesticides that are applied. And actually the same thing occurs in cancer. So anti-cancer drugs may initially be effective, but then later cancer cells that are resistant can make the cancer come back again. Now we come to population genetics. So it's important to understand that individuals do not evolve, only populations evolve. So evolution is a population level process. It's one of those emergent properties that I talked about at the beginning of the course. It only shows up at the population level. And in macroevolution, the frequency of alleles changes. But if evolution's not occurring, then the allele frequencies stay the same. And if the frequency of alleles doesn't change from one generation to the next, that's called Hardy-Weinberg equilibrium. So in the case of Hardy-Weinberg equilibrium, the allele frequencies are staying the same generation after generation. So, unless certain factors operate. And so what are those factors? There's 5 of these. These are sometimes called the 5 agents of evolution. And the first one is a small population. So if a population is driven to near extinction, that will influence the allele frequencies, because when the population is very small, the frequencies of alleles are no longer statistically random. There's statistical fluctuations that occur, just like if you were to flip a coin a few times, you get some unusual results, but if you flip a coin 100 times, you should get very close to 50-50 heads and tails. But so in a small population, the frequencies of alleles can change, and that's also called genetic drift. And that's an important term to keep in mind. The genetic drift is just having a small population and thus allele frequencies that are changed. Another is called gene flow. So gene flow is immigration or emigration, that is individuals coming into a population or leaving a population can also change the allele frequencies. And another one is mutation. So if mutations occur, that can change allele frequency. The fourth one is non-random mating. So normally mating is random, but if there's inbreeding, then the mating is no longer random because the allele frequencies of closely related individuals are going to be different than those of a random population. And so non-random mating or inbreeding will affect allele frequencies. And the last one is natural selection. So if there's natural selection occurring, that's going to change the allele frequencies. Chapter 14 is about speciation. This is how do new species arise. And if new species arrive, first we have to define what we mean by species. So the definition of species, one possible definition is the biological species concept. And this is the idea that species consists of mating individuals that have the potential to produce fertile offspring. So in order for them to be the same species, they have to produce fertile offspring or be capable of that. So for example, you can mate a horse and a donkey, and that gives you a mule, but the mule is sterile, and so the horse and the donkey are not the same species. They're considered different species because when they mate, they do not produce fertile offspring. Another form of reproductive isolation is temporal isolation, and this is when two populations of plants, for example, become different species by pollinating in different seasons. So if two populations evolve so that they reproduce at different times of the year, that's another way that they can become reproductively isolated. And another important concept is adaptive radiation. This is the idea that you can get numerous species from a common ancestor if they are somehow separated. And one example of that is the birds on the Galapagos Islands called the finches. So these islands, which I showed earlier off of South America, have many different species of finches that evolved on the separate islands. And genetic evidence shows that they're all related to each other and had a common ancestor, but as they each moved to a different island, they evolved into different species. And lastly, then there's different forms of reproductive isolation. So we've already looked at geographical isolation and temporal isolation, but there's also three others. There's habitat isolation, and this is This is similar to geographical, but in this case, they're separated by different environments. So lions and tigers are capable of mating, but they don't normally do so because the tigers are living in a jungle and the lions are living in a more of a savanna type environment. So they live in different environments, and so they don't encounter each other. And another one is behavioral isolation. So this is often observed in birds which have elaborate mating rituals. And those mating rituals tend to isolate them in different species, because over time they don't mate with birds that are not having that same ritual, and so the mutations accumulate and they become different species. And the last is called mechanical isolation, and this is basically the idea that the reproductive organs may not be compatible because of differences in shape or size. Chapter 15 is on the subject of macroevolution. So macroevolution is major changes that occurred in the history of life. So we're talking about the whole history of life on Earth, and some events were very major events that had large influences, as opposed to microevolution, which was the smaller changes. So first of all, we have to understand how scientists are able to determine the age of fossils. And this one way to do this is called radiometric dating. So let's look at one example first of carbon, and then we'll look at some other isotopes. So carbon, as we saw earlier in the course, has the stable isotope carbon-12, and this is unchanging, but it also has carbon-14, and carbon-14 is radioactive and therefore unstable. So over time, carbon-14 breaks down, and when a plant is alive, it's doing photosynthesis, so it's bringing in CO2 gas and forming sugar out of it. But in some of that, the carbon in the CO2 is carbon-12 and some of it is carbon-14. And so there's a ratio of the two isotopes of carbon in the plant. And while it's alive, that ratio is steady and unchanging. But then after the plant dies, the ratio changes, it goes lower. So the ratio of carbon-14 and carbon-12 is going to decrease over time, because the number in the numerator gets smaller and the number in the denominator does not. So what is how fast does carbon-14 break down? That's called the half-life of carbon-14, and the half-life of carbon-14 is 5,700 years. What does this mean? It means that this is the amount of time it takes for half the amount of carbon-14 to break down. So imagine you have a pie, and imagine that you cut the pie in half, and imagine you give away half of the pie. So you cut it in half, remove half of it, that half that was removed is the half that breaks down. And so that takes 5,700 years. Now you've got half the pie, and now let's say you cut it in half again and remove another half. So now you have a quarter left. has 2 half-lives. And let's say cut what's remaining in half again and remove half. That's a 3rd half-life. So after 3 half-lives, you have 1/8 remaining. And what is 3 half-lives? It's 3 half-lives would be 17,100 years. Just this number times 3. That's 3 half-lives. And the limit for carbon-14 is going to be about 75,000 years. So that's the amount of time that you can use carbon-14 to date things. It's not very much compared to the history of life, which is about 4 billion years. So we can't use carbon-14 for very old fossils like dinosaur fossils, something like that. It can only be used for recent things, such as archaeological remains. But what about older things like fossilized dinosaur bones, for example? We have to use different isotopes. There's one that you can use is called potassium-40. Potassium-40 has a much longer half-life, and so you can use that one to date older things. And for very old things like rocks that are very old that you want to date, you can use uranium. Uranium is a radioactive element, so it's unstable. It ultimately breaks down into lead. So by measuring the ratio of uranium to lead, you can determine the age of very old rocks, and this is how the age of the Earth was determined, was by measuring this ratio. So during the history of life, we have different stages, different periods and eras. And 2 of the major eras are the Paleozoic and the Mesozoic. And the Paleozoic ends with the Permian mass extinction. And the Mesozoic ended with the Cretaceous. Mass extinction. We don't know as much about the Permian mass extinction, but the Cretaceous mass extinction, there's evidence that it was caused by an asteroid hitting the Earth. And the evidence for that is that at the boundary between the Cretaceous and the period after it, it's called a KT boundary. At that boundary, there's a layer of iridium. And iridium is an element that is common in asteroids and meteorites, things in outer space, but not as common on the Earth. And so the level or concentration of iridium at the KT boundary was much higher than ordinary. And that's one piece of evidence for the Cretaceous mass extinction being caused by the by an asteroid impact. Another is that a large crater was found off the coast of Mexico, and that's another evidence for that. Another important concept is the concept of derived characters. So these are characteristics that distinguish different groups within an evolutionary tree. So for example, we can look at the mammals, and in the case of the mammals, the platypus is distinguished from other mammals by a derived character called gestation. So in the mammals, you have, here's an evolutionary tree comparing different vertebrates. We have the frog, and then we have the iguana, and then we have mammals. And this is figure 15.16a in the textbook. So the frog is distinguished from other vertebrates by the fact that its eggs are laid in water. And the eggs of other animals, such as lizards, are not laid in water because they have, their eggs are protected by a membrane called the amnion that is a kind of watertight membrane that keeps the egg moist. So that characteristic, the amnion, is an example of a derived character, and it allows us to, these derived characters allow us to assemble or build an evolutionary tree. So the platypus is different from other mammals because unlike the other mammals, the platypus lays eggs. It's the only mammal that lays eggs, but the other mammals have gestation, which means that the embryo develops inside a uterus. And then there's different kinds of mammals. There's the marsupials, which include the kangaroo, and these part of the embryonic development occurs outside of the uterus in a pouch. And then you have all the other mammals that have a long gestation in the uterus. So you have these different derived characters, and from that you can build an evolutionary tree. And this is one way to build evolutionary tree. Another way is by looking at DNA sequence and looking at how different or similar DNA sequence is. And DNA sequences that are very different, such as frog versus mammals, would be having a lot of differences in DNA sequence, whereas the frog and the iguana might have fewer DNA differences in their DNA sequence. So evolutionary trees are a way to represent evolution, and evolution is best understood as a tree rather than as any kind of progressive process. There is no direction to evolution. It's simply mutation and then the branching off of individuals into different species, and so evolution explains both the diversity of life and the unity of life. So it explains the unity because we can see that very different organisms are derived from common ancestors. And it explains the diversity because it shows all the branch points, that it's a very branching tree-like arrangement. And finally, in chapter 15, we have the taxonomy. This is the way in which living organisms are organized and put into different levels and named. So the name of an organism is a binomial name where they have first the genus name and the species name. For example, humans are Homo sapiens. Homo is the genus and sapiens is the species. So each species is named by that binomial approach. And the different taxonomic levels are shown here. So the largest level is kingdom, and these are the plants, the animals, the fungi, etc. And then you have the phylum. So phylum, each kingdom is divided into different phyla. So for example, in the animal kingdom, there's different phylums that represent the different body plans. So one phylum is the animals with bilateral symmetry, and then there's other animals that have radial symmetry, such as the starfish would be an example of radial symmetry. So they'd be in a different phylum from the bilateral symmetric animals. So that's just one example of different phyla. And then within phyla, there's further division by class, and then the classes divided into orders, and the orders into families, families into genus, and then into species. And there's an easy way to remember this. You just remember this sentence: King Philip came over from Greece singing. So the first letter in each word corresponds to each level here. So you just remember that sentence